All right, so it looks like everyone's getting transferred in from the waiting room. Thanks for your patience. We had a little bit of a technical difficulty, but we are ready to go. So welcome to our COVID-19 webinar, where we'll be walking through testing and clinical discussion. And today we have Dr. Shippy with us, and I'll introduce her in just a moment. Before Dr. Shippy talks, we're actually going to be able to hear from Krishna Murthy, who is Vibrant's Director of Biomedical Engineering. And he is not only one of the smartest people at Vibrant, which is, if you know Vibrant, that's a serious compliment, because we're just full of incredibly smart people. Hari is actually one of the founding members of Vibrant. He's been with the company since its inception. He's part of the core team that actually developed the microarray technology, and that included across peptide, protein, and DNA array platforms. So he's going to be treating us to a discussion about Vibrant's technology. I know there's lots of questions about that. And he is the man to talk to when it comes to our technology. So he oversees that, plus the key products that Vibrant brings to market. He is part of our scientific advisory team. He ensures our third-party validations and verifications of Vibrant products, as well as he's also published in peer-reviewed journals. So I'm going to hand it over to Hari in just a second. I also want to introduce you to Dr. Shippy, because she's going to jump right after Hari is done talking. So Dr. Shippy, welcome. Thanks for joining us today. I really appreciate your time. A little bit about her. She's on a mission to educate and help people across the world live the healthiest, most optimal lives possible using cutting-edge science, research, and genetic information to find and treat root causes. She specializes in environmental specializes in environmental toxicity, preconception and reproductive wellness, and mold exposure. And so she uses a lot of epigenetic and genetic information and works with the body's ability to express or repress helpful or detrimental genes in that process of healing and reversing certain illnesses. She was an IBM engineer before becoming a doctor. She attended University of Texas Medical School. She's board certified in internal medicine and a certified functional medicine physician, and she practices in Austin, Texas. She's also authored a couple of books and is currently working on a third. There is her website if you want to find out more about her or get in touch with her, AnnShippyMD.com. And now that we've run through really quick, Hari, if you are on the phone, I'm going to go ahead and turn it over to you. And let me know when you want to advance slides, and I'll just click them over for you.
Sure, perfect. Thanks, Sarah, for the introduction, and welcome everyone to the webinar today. Thanks for joining us. To get started, I think it'll be fair to give you an introduction to the company itself. We here at Vibrant, we were founded by Dr. John Rajashekaran. He's a pioneer in the field of peptide array diagnostics. He's one of the most persistent scientists out there. Sticking with peptide array and making understanding the nuances of really making array technology work. He's been in this field for a long time. Has plenty of patents in the peptide, protein, and DNA microarray space. He's the thought leader and really keeps the troops going here at Vibrant. More about the company. The company itself was founded in 2011. We started out with developing our chip technology and filed a lot of our initial patents on the technologies that we developed here at Vibrant. And the first 5 years were mainly development of the technology and also validating it and making sure that the new product that we are bringing to market is well validated and matches with the current set of diagnostics that is already available in the market. So we had a lot of industry collaborations to do that. Mayo Clinic, some of you who've read our publications in the past would be familiar. We work with Mayo Clinic on celiac disease. We are working currently with the University of Washington on bringing a better, rheumatoid arthritis product to the market. Columbia University on Lyme disease, and there are several other collaborations that we work on. You should be able to find more information on the publications and studies that we are doing on our website under publications. So yeah, we basically developed this microarray platform and did industry collaborations to validate it. And we found it optimal to start a lab. We started a CLIA and CAP certified lab, which is Vibrant America. That is for mainly proteomics tests and protein chemistries. And we also have a lab in Vibrant Genomics, which does genetic testing. Both of our labs are CLIA and CAP accredited, and we are located in San Carlos, California. And the chip manufacturing itself, the microarray plates and other things that we manufacture, they are manufactured in ISO 13485 facility, which is located in Belmont. And ISO 13485, as you people know, is one of the most rigorous manufacturing standards that's set for medical equipment, and we are compliant to that. Sarah, if you could, move on to the next slide.
Okay.
When COVID initially started spreading in the US, we wanted to make a contribution with our microarray. A couple of advantages that we have, which we'll discuss in the subsequent slides as well, is we have the ability to multiplex and test for multiple antigens at the same time. And, we wanted to really make a contribution in this space. So if you look at antibody tests in general, they are done on something called as a ELISA platform. So what really is the ELISA platform is what we're looking at here. So on the left, you see a 96 well plate. It's basically like mini tubes inside of a plate. And you can run 96 reactions at the same time on a single plate. And what typically is done in the manufacturing of these plates is each of these wells is coated with a particular antigen against which you are checking for the antibody. So you see, the graph on the right-hand side, you see a solid plate which represents the base of a well plate. And there you have an antigen that is adsorbed on that surface. So you have an antigen coated 96 well plate. And to this, each of these wells, you will basically add a serum sample of the patient you are testing. So that is the primary antibody coupling to the antigen. So the primary antibody is the antibody that's present in the patient's serum. And when you add it to each well, if the patient has an antibody to the antigen, it's going to go and bind to the antigen. And what happens after this step is typically you have a wash step, which cleans out any unbound antibodies that are there. And you then add a secondary antibody, which has an enzyme at its Fc region. So what this does is it enables labeling of the primary antibody that's already attached. And as a step 4, when you have a chromogen substrate that's added, it gives out a particular light, which will indicate that there is a primary antibody that was detected, which has been bound to the antigen. So this is the typical ELISA workflow for antibody test. This has really been a helpful method. This is a pretty old method as well. So it has a set of disadvantages which we focused on and improved when we brought out our microarray. So main disadvantages, you cannot test for multiple antibodies at the same time. The main reason for that is, each well, you can coat it with only a single antigen. So if you have to really test for multiple antibodies, then you will have to have multiple plates. So if I have to test for something like 4 antigens, then I need to have 4 plates, and I have to add the patient's serum in 4 different plates, and then detect all the 4 antibodies for that given patient. That is, as you know, is going to be very expensive and laborious process. And also, since this was developed a long time ago, the substrate itself that is used in many of these 96 well plates, the density of the antigen that is coupled to the surface will be low. And that makes a big difference. The reason being, the more dense you have antigen on the surface of your well plate, it's going to be able to detect lower level of antibodies. So your sensitivity is going to suffer if your substrate is not dense enough. So that is one of the other disadvantages that are present in the ELISA-based test detection methods. So we looked at these issues and we solved them and addressed them in our microarray platform. If you could move on to the next slide, Sarah, we could show how our microarray platform is manufactured. So this is called the vibrant filler plate. So basically what it is we made something that's complementary to a 96 well plate. And what I mean by that is when you stack a 96 filler plate on top of a 96 well plate, it's going to basically merge in, and the filler is going to go inside the well and touch the liquid, whichever is there inside the well. Let's go over the the manufacturing itself of the array. At the top left, you see a circular disc. It looks similar to your compact disc that we used to use a long time ago, right? So that circular disc is silicon wafer, and what we do is we coat this complete silicon wafer with the protein of interest. So say for instance for COVID, we have vibrant tests for 4 antigens, right? So we would be using 4 wafers, and on each wafer we will be coating the protein of interest. So S1 protein on a wafer, RBD protein on a different wafer, S2 protein on another, and then the nuclear protein on another. So we'll have 4 wafers which are coated with the protein, and we will take it through something called wafer dicing. So what this does is, this is a high precision wafer dicer which will cut the wafer into very small chips. And that's what you see subsequently shown there, where you see the lines and the mini lines that are there on the wafer. That's basically the dicing machine cutting the silicon wafer into very small chips. And once the wafer is cut into small chips, we have something called as the chip sorting. So the S1, S2, RBD, and the nuclear protein, all these different chips that are there in 4 different wafers, we tape it into something like yesteryear audio tapes. We have these chips picked and placed into a roll, and this essentially is where what we use to store the chips before manufacturing and making the filler plate. So you have at the end of these 2 steps tapes on which the chips are already placed, and they have their antigen coated on them. So this is one. And if you look at the bottom left of the screen, there you see the filler plate, the physical filler plate itself. What's here is a 24 filler plate. What we use for COVID is actually a 96 filler plate, so it's a lot more dense. But the idea is the same. So you have a filler plate and you have an epoxy dispensing machine, which puts a small dot of epoxy, which is basically glue, on each of these fillers. And after the epoxy is dispensed on the fillers, it goes to a pick and place instrument. The pick and place instrument is kind of like what puts everything together. So on one hand, you have the epoxy dispense filler plate, and then the other you have the chips which are on tape. And the pick and place instrument takes the chip from the tape and puts it on the filler plate. So at the end of this process, you will have a filler plate which has our reference chip. I'll show you a picture of exactly how our COVID testing plate looks like. But at the end of the process, it's basically the filler plate with the 4 chips that are there for testing the particular antigen. If we can move on to the next slide, Sarah. Thanks. So you see here, the bottom left is the 96 filler plate with the 4 chips in the center. You also see a single shot of a single filler, how the chips are placed on it. You see it's labeled clearly for S1 glycoprotein, S2 glycoprotein, RBD, and nucleoprotein. So those are the 4 antigen-coated chips that are there on each of these fillers. So when you put this 96 filler plate on a 96 well plate, you could run 96 serum samples which are diluted and placed inside of the well plate at the same time. So the serum sample which is there in the well will come in contact with this filler, and the filler has your 4 antigen-coated chips on it. So you are multiplexing and detecting the antibodies for 4 antigens at the same time. This is important concept to get in, because this really allows us to do the multiplexing, and the workflow is so much simplified. Because we have the ability to do this. And the chips are manufactured in ISO setting, and what we do to actually run the assay is also highly automated, because Vibrant is always looked at minimal handling with humans, because I think the platform itself lends itself to automation. And we use Hamilton liquid handlers, Microlab Stars, Vantage instruments to do our assays. And these are pre-programmed on these instruments. And we load the serum samples or eluted, dried blood spot samples on the tools. And then we have the filler plates also mounted on these tools. And the assay is, performed automatically. And the great part about it is, we have integrated assay and scan station. So it's you load the samples, you load the plates on which the samples are going to be run. We have the buffers that are placed on the instrument as well. And what happens there is the assay is automated, and then the filler plate is taken automatically and placed on the scan station, and the scanner scans the each of these fillers. The signal that's coming out of these chips is read by the scanning instrument and translated into numerical values, which then are compared with thresholds, lending itself to giving out the results whether a particular sample is positive for an antigen or not. This automation also enables us to have very high reproducibility, because there's very minimal change in how each sample is being handled here at Vibrant. We also are able to run a large volume of samples because of this. Current capacity for COVID is running 50,000 samples per day, and this can be easily increased as well with more tools. Next slide, sir. So what we've seen thus far, the filler plate itself with the multiple antigen testing. The table here actually shows why we are the only lab doing 4 different antigens, because we have this proprietary platform in which we are able to put multiple antigens on the same well. We are able to run spike S1, spike S2, RBD, and the nucleoprotein, versus the other tests that are there in the market typically test for a single antigen. It's either the nucleoprotein or the spike glycoprotein. So I think that's this is mainly enabled because of the technology that we've developed here at Vibrant. So on this slide here, we look at the sample report. This is typically what you could expect if you run the sample of your patient here at Vibrant. We have the patient information, the provider information right on top. And we also clearly specify, what sample type was sent in, whether it was serum or dried blood spot. The EUA approval that Vibrant has, we are the only test that has been EUA approved for, dried blood spot collection and serum. And that's something that, will be a great enabler for practices, because there is no other test right now that's FDA approved which can accept dried blood spot as a sample type. So going back to the table, so you see here the spike glycoprotein S1 receptor binding domain, spike glycoprotein S2, nucleoprotein, and then you have 2 columns, IgM and IgG, and it's going to clearly say whether it was positive or negative for each one of those. And below the table, you see a summary interpretation, which will give you a brief idea of what that really means. Technically, it's basically indicative of evidence of infection if any one of these antibodies are lighting up. Next slide, Sarah. So results in interpretation, like I said, any one antibody that's lighting up, it shows evidence of infection. And there are tests out there that are checking for IgM antibodies as well, but I don't see any test that reports them separately. So like you saw in the sample report, we report on IgM and IgG separately. We are not a total antibody test. We don't do everything together and just say positive or negative. We are individually giving you the results for IgM and individually giving you the results for IgG. So the importance of that is, typically IgM is your early antibody, so you're able to say whether it's a recent infection, and that will definitely help with contact tracing and who should be in quarantine and things like that. And your IgG positivity, of course, indicates your exposure to the virus. Next slide, Sarah. So wanted to take a minute and actually go over dried blood spot, because we, like I said, we are the only dried blood spot antibody test that has been EUA approved. This is a big advantage for practices because you don't need a separate phlebotomist. Any medical personnel that you have on site can assist and help collect the dried blood spot. And we validated the accuracy of this, and the data of that is the coming slides. Basically, we were able to find 100% clinical agreement between a serum collection and dried blood spot collection. And we worked with very good hospital, Gunnison Valley Hospital. Shout out to them for really helping us with a lot of sample collection in the initial days. So serum and dried blood spot collection comparison, we are having 100% clinical accuracy. One more advantage of TBS, of course, is because it's in a dried format, it's much more stable at ambient temperature, which is why we are able to ship it at room temperature. We did shipping studies, and we were able to see that it's stable for more than 7 days, which is very hard to imagine with samples from something like a EDTA collection or something like that. At room temperature, it's very difficult to ship. But dried blood spot stability at ambient temperature, I think, is fantastic. We also ran a position and reproducibility study. So what we mean by that is, if you see the dried blood spot here, we have 5 different spots and collections on each of those. If we punch and collect the blood from each of those spots and run them separately, we are still able to see that it gives the same result. So it's very precise and reproducible. Now, having said this, dried blood spot, there's a reason why we were the only test which was able to get the dried blood spot approved. It's because we brought a lot of rigor to the sample collection, and this is a very important take home for practitioners who are planning to bring on the dried blood spot collection at the facility. It's very, very important that the collection method utilizes some of the key points that I'm mentioning here. There'll be a separate webinar on the dried blood spot collection that we'll be bringing out. But some important things that I wanted to mention in this is it's very important to rinse your hand in warm water for extended period of time so that that good blood flow to your fingertips. Typically, I know, and I'm guilty as well. When I'm given a kit, I would probably just go and write, prick it and collect the blood. But it would be a big issue if you were to do that. So rinsing in warm water really stimulates the blood flow, and it will really help you in collecting your blood spot, right? And we've also seen that holding your hands below your waist will improve the circulation as well. So rinsing the hand in warm water for more than 1 minute or 1 minute, and placing them above your waist, below your waist, and milking the blood out. I think that's a very important step. We should also make sure that we are not literally touching the card with the spot of blood. So you want to form a drop of blood on your hand and let it drop to the paper below, like shown in the diagram here. The blood should form in your tip of your finger and it should drop on the plate. You should never touch the dried blood spot directly with your fingertip, because that's going to stimulate more blood flow to it, and you don't want that. We also want only a single drop placed on each of these circles. So you let a drop form and drop a single one in each of these circles and fill up. I think the maximum number of circles that you could fill before drying and then shipping it out to us. So drying of the card is also equally important, because you need to place it in a place that's well ventilated and dry it for about 2 hours before shipping it to us, so that the blood sample is very stable. So these 3 takeaways, it's very important to remember: rinsing with warm water, holding the hand below the waist, and stimulating the blood flow, not touching the DBS card, and only letting 1 drop of blood in each circle. These are all very important points to consider before shipping the cards out to us. If we could move to the next slide. So, moving on, we want to talk a little bit about the validations we have done. Our complete validation, as you know, Vibrant is a very transparent company. We have the complete validation that we've done on our website. Under COVID-19, you should be able to find it. But here are the key takeaways. Within the analytical study, we did persistence and reproducibility, which is very important. That the same sample, you run it multiple times, it should give you the same result. That's technically what this looks for. So what we did is we ran 11 samples with 2 replicates twice daily over 20 days, and we also ran 5 samples with 5 replicates over 5 days with 3 lots. So each time we manufacture our chip, it's considered a lot under ISO 13485. So we manufactured 3 different lots, and we did a lot-to-lot comparison. How much is the variation in the antibody level for S1 IgG, S1 IgM, RBD IgM, IgG, and so forth. So you see the second column shows the lot-to-lot PV. So there's about 5% variation between the lots. That's a really high standard, and we are able to achieve it primarily because of the complete automation in our processes. And persistence, of course, is running the same samples over and over again, and the variation there you see is between 7 to 10% is what we see for all these different antibodies. So after persistence and reproducibility, of course, we went on to do the interference study. There are a lot of substances that are present in your blood which could interfere with the antibody test. So what we did was we spiked the interference agents that are shown here, the bottom left of your screen. Bilirubin, your triglycerides, hemoglobin, even azithromycin, which is one of the drugs that was initially approved by the CDC FDA. These are all spiked at the concentration that's shown here, and we did not see any interference at these concentrations. So this was very encouraging because drug interference to antibody could impact the antibody levels, but we did not see any of that interference in our assay. We also did the cross-reactivity study that you see on the right. These are all samples that were positive for using a DNA test, like upper respiratory panel. The serum samples from those individuals were run on our coronavirus chip, the 4 chips S1, S2. And we see that we did not have any false positives. And that's a very important aspect of the test, because that speaks to the specificity of the test. So when you have 0 false positives, your specificity of the test is really high. You see here all the relevant infections like influenza A, B, HCV, HBV. they did not have any false positivity here. More importantly, if you look at the middle of the table, you will see other coronaviruses, alpha and beta coronaviruses, which are very similar to the SARS-CoV-2. We did not see any false positivity for those samples as well. So it really speaks to the specificity of our assay. And moving on to the next slide, we look at the clinical studies, and I think Dr. Shippy was involved in a lot of this, and she's going to take over from here, and I'll chip in as necessary. Dr. Shippy, if you could go over these slides.
I would love to, and I just want to tell you how grateful I am for how the Vibrant Laboratory has been helping me to manage my patients. Know that they are indeed infected, and then to find out when they are no longer going to affect the people around them. So I'm very, very grateful for all this amazing work that you guys have been doing. So, and what I love is the rigor that you guys have brought to really making sure that this is both the serum test and the finger prick are, really going to help us to give the best medical care that we can. So in this, looking at the clinical studies to show how good the test is, what Vibrant did is they took 225 clinically positive by PCR, and then 296 that were negative by PCR, as well as an enormous number of people that were healthy prior to the outbreak. So you wouldn't have expected anybody to have been infected unknowingly, have been asymptomatic. And then also comparing it to people that have other infections like Lyme, CMV, hepatitis, to make sure that there isn't any confusion between the different disorders. So almost 5,000 samples. And what the study showed is very helpful for us as clinicians to help to determine when the best time to test is, and to know how good the test is in different time frames. So in the lower left corner, you can see by time frame. So less than 7 days, the sensitivity. So just a reminder, sensitivity helps us to know what percentage of people that we capture that are actually positive, and sensitivity helps us to know, are we going to have a positive test when it's really not positive, so the false positives. So in less than 7 day period, still pretty good detection, so 89, almost 90% sensitivity and 96% specificity. So that's really excellent. But then when you get to that 7 to 14 day period, it's 100%. And so we can really trust our results when we're in that time frame, and it's still very, very good after that. So what happens though is after 14 days, the IgM levels start tapering off for many patients, so it might not show an IgM level. So when we look at all of those samples together, really, really great sensitivity and specificities, and you can see that up over in the right-hand side with the IgG overall. With the positive patients, it's a sensitivity of 98.67%, and then the negatives, a specificity of a 99.76. Same kind of thing with the IgM. And so when you combine those two together, it's really impressive. There's a 99.56 sensitivity and a specificity of a 99.76. And I think that's what we're really all shooting for, because we want to make sure that we really identify patients that are positive, but then we don't want to have positives come back for patients that really aren't, because that has such enormous implications for their life. So I think I'm really, really happy with both the sensitivity and the specificity of doing this test to really help augment what I'm doing with the PCR. Okay, do the next slide. And then, I've done a lot of the serum tests. as challenging as it has been to, when I think people are positive, you know, to be able to either draw them safely in my office or to have another draw station. So I'm really excited what's been shown with the comparing the serum test to the drugged blood spot performance. So what this shows is the rigor that was done, and those I'm sure to show to you know, Vibrant themselves, but also to the FDA, is that it's really an excellent correlation. So in the matrix equivalency study, you want you know, looking at the data together, a slope of 1, an intercept of as close to 0 as possible, an R of close to 1. So what we can see is that the two, the blood spot and the serum, correlate really, really well. So then in the blood spot, they showed a correlation of exactly 100%. It's 1-for-1. So that's going to make it so much easier for us to get these excellent results with our patients without having to either have a nurse or phlebotomist in our office or find somebody who will draw them safely. We can easily, you know, pretty much any office can administer a blood spot. So this is super exciting. Okay, next slide. Ready for the next slide? Okay, so what I love too about the data that's collected through these studies that were done for the correlation is to look and see what, you know, on average happens with the antibody levels. So the IgM is in orange, and we can see that over, you know, between week 1 and week 3, the levels of IgM generally for most people tend to go under the radar, and then the Ig is building up over the first 4 weeks and then kind of leveling off. So in, you know, they have done several of these cases where they've looked at them at the levels with the PCR and with the antibody levels over several weeks. So the first patient was 51, female, with fever, chills, muscle aches, shortness of breath, and nausea. And, you know, so kind of a traditional symptom experience for the patients. And what we can see is the first sample was positive for PCR, IgM, and IgG, continued through weeks 2 and 3, but then on the 4th week, the patient went negative for PCR and for IgM, but continued to have the IgG antibodies. Then in case 2, this was a 47-year-old male patient who had fever, chills, nausea, the loss of taste and smell that we're seeing in so many patients, and then fatigue. So in this patient, there was a little bit of a different scenario. So time 1 was positive for PCR, IgM, and IgG, but then when the PCR went negative at the second time frame, the IgM was still a little positive, and but then had switched over to the positive IgG. And then finally on the 3rd time was negative for PCR and IgM, and still positive for IgG. And this would correlate really well with the patient that I used the serum test for, that really helped me to figure out when it was safe for him to come out of quarantine. He had a really severe episode of the coronavirus. He was hospitalized for a few days until his treatment really kicked in. And then, but we had to really make sure that he was not contagious anymore, because his wife had, her mother had stage 4 lung cancer, and she was rotating with her siblings to take care of her mother. So we wanted to make absolutely sure that before they broke quarantine that he was truly negative. So doing these serial tests really helped for me, and he did stay positive with the IgM after he was multiple times negative on the PCR. So we were, but it took a good between 5 and 6 weeks for him to be able to come out of quarantine. And then case 3 is a 58-year-old patient who didn't have any symptoms, was positive on PCR, IgM, and IgG antibodies on the first test, but then became negative for both the PCR and the IgM at the same time, and also continued positive with the IgG antibodies. Okay, next slide. Okay, you can take it back.
Sure, Doctor. Thanks for that information. Now moving on to how you could order the test and the pricing of the test itself. What you could do is any provider who already has an account with us, they can just go to their login and they should be able to see the COVID order right there, and you will have the option of ordering the test right in your login. If you are a new provider and you want to open a new account with us, then there's a new account form that is available. This is a hyperlink, and subsequent to the webinar, we could also send out an email with the link to open a new account form. Basically, if you have a set of information that you'll need to input your name, NPI number, etc., to form a new account with us, and then you will be good to order. The pricing on the test itself is $199, and I want to clearly show you what you're paying for here. So, typically, the tests that are run out there in the market is for a single antigen and for a single subclass of antibody, and it's priced at I think about $120. But here at Vibrant, we are doing tests against 4 antigens and 2 subclasses of antibodies. So effectively, it's 4 times 2. You get 8 data points on the same patient for the same price. And it's a EUA approved test for BBS and serum samples. So I think it's really a no-brainer. For value, I think this is the best test out there, and the price that we are offering it is very competitive. Next slide. Oh, one thing I have to mention here is, we also provide the super bill. So your patient can use the super bill to, send it to their insurance company and get reimbursed for the test.
Well, and can I just say
so
one
slide
thing
here
is I think it's really helpful to have all 4 antibodies, because not everybody makes antibodies to everyone. So to really not miss somebody that's positive, it's really helpful to have all 4. Exactly
right. So, the way we think of it is, when you cast a net, you cast it as wide as possible. So to get every patient out there who has an antibody. We had some, patients who called us, who are RT-PCR positive, and we're very thankful that they were able to see the antibody positivity through Vibrant tests that they were not able to see in other tests in the market. So, it's basically all the key functional proteins of the coronavirus. We have it multiplexed onto a single pillar. So I think it's the test to beat in the market. Some mandatory disclaimers that we have to put on the report. We have to be very clear that this is a EUA approved test, and EUA is emergency use authorization. So it's an authorization, but not an approval. So we are working with FDA to have a 510(k) on this as well in the future, and we are also working on a home-based white blood spot collection, but we don't have EUA for that. But that I want to reiterate that this is a EUA approved test, so it's an emergency use authorized test, but not an approved test. This test is authorized only for the presence of antibodies against SARS-CoV-2 and not for other viruses. So this is the set of disclaimers that you see on the report as well, and it's mandatory for any person who is EUA approved and is offering this antibody test. Next slide, Sarah, please. The key takeaways we have in the last 2 slides. Again, to reiterate, we are the only test which is authorized for IgG and IgM antibodies against the 4 key antigens of the novel coronavirus. And we are the only test which has a dried blood spot collection, which greatly simplifies the ease of collection at, the clinician site. We also test for IgM and IgG antibodies, and we report on them separately. So you will know if a infection is, recent, if the IgM antibody is present, and the exposure is the exposure happened a long time ago if it was just primarily IgG that you see on the patient. Next slide, Sarah. Like I said, to summarize, we are the only EUA approved test for dried blood spot and serum collection. 4 antigens, S1, RBD, S2, and the nucleoprotein are tested at the same time in a multiplex fashion. We individually report on IgM and IgG, and at a great cost of $199. That's pretty much the summary of the test that we are providing here at Vibrant, and I think we could throw it open for some questions before wrapping up here, Sarah.
Great. So if you do have any questions, feel free to type those in to the Q&A. It looks like we have a few ready. Dr. Shippy, did you have anything else that you wanted to add while I'm kind of going through to take a look at the questions that we have in here?
No, I think this covers it pretty well. We're not really getting into prevention and treatment. I would just say if anybody's, I've treated a number of patients now, so if anybody would like information on what I'm finding works to help with treatment, I have a webinar that I did a few weeks that's in the blog section of my website at annshippymd.com, and really nerd out on what the mechanisms of action are and that kind of thing. That's really helpful for practitioners that are wanting to try to help their patients with the best information that we have at the time. And then the IFM website, it's a functional medicine website, it's ifm.org. They have a lot of material that's very helpful about things that will help support the immune system to do a better job at fighting off the virus. And then I just really think, I'm hoping that we kind of change the conversation that we're having about health in America, that we use this as a period of time to really help people to be healthier and more resilient, and to be doing things on a daily basis that really help them to be healthy, so that they're less susceptible to this illness, as well as all the other the way that I look at it, pandemics that are happening in our country, from diabetes and cancer and autoimmune disease. I think this this pandemic is just that, it's showing the tip of the iceberg that we really need to be working on these illnesses in a different way, rather than just managing chronic disease.
Great. Let's see. I think there's a few questions. Some of these. So Claire, there actually was a slide that showed the sample report. These slides will be made available to download in our portal once the recording is live. So you can actually log in and access those there. You can see what the sample report looks like. But there's also, if you already have an account, your territory sales rep will also have a sample report that you can take a look at. And let's see. Christina asked, can you clarify, are you recommending that patients self-quarantine while IgM is still detectable?
Oh, that's a great question. So what I did with this patient, that it was so important for him to, be absolutely certain that he wasn't contagious anymore, was we looked at serial Vibrant tests. And what we could see is that his IgM, the number of IgM antibodies were going down. But a couple of times his PCR was still positive. So at the time that his PCR was negative, we could also see that at that time there was also IgA reporting and IgM. We could see that they had considerably gone down. So I think it's nice to really combine the PCR testing with the antibody testing to see what the patient's immune system is doing. So we didn't wait until his IgM was completely negative, but we could see that it was going in that direction. And unfortunately, his wife never, we confirmed with the Vibrant testing on her a couple of times as well to make sure that she hadn't accidentally got it. They actually did a really great job of quarantining within the house. So she never had any antibodies, no PCR. She did great.
Awesome. Let's see. Ira had a question. Does bilirubin interfere with the test? And how about someone with Gilbert disease?
Yeah, so for bilirubin, we actually did the study in interferences, and we spiked it at a very high level, and we did not see interference with bilirubin.
And Dr. Shippy, do you know about Gilbert's disease as far as would that interfere with the results?
It shouldn't. If they looked at since you guys looked at the bilirubin, it should be accurate.
Okay, great. Let's see. Margaret asked, do any other labs check for 4 spike proteins? Which, so Margaret, there actually was a slide that showed what our test measures and then other labs that are available. So no, we are the only one that's measuring for all 4 of those antigens. If a patient is negative for IgG but suspects past infections, would a Vibrant test show more antibodies?
That would be absolutely.
Sorry, could you repeat that?
Yeah. So if a patient's negative, if they've done one of these rapid tests or one of the other tests and they were negative, but they're pretty sure they had it, definitely the Vibrant test is more likely to show it. There may be a case like me where I have a history of my IgG running a little low. So when I tested myself, I was positive for IgM a couple times, and then I didn't convert to IgG, but that's because I have a bit of an immunodeficiency that I just don't make the IgG effectively.
Great. And and Dr. Shippy, that's one of the most commonly asked questions about does IgG confer, immunoprotection? And I think that, from what we know is we don't know. That's one of those sort of unknowns with this particular virus. But an important point that you make there is there does appear to be a small subset of people that do not seroconvert from IgM to IgG. So as far as interpreting the report, can you speak a little bit about because one of the questions is, how do we interpret this, letting patients know they're no longer infected, or, what if we don't see this resolution in antibodies? What are some, so you mentioned IgG deficiency could be a reason why somebody might not see IgG antibodies produced, especially upon multiple tests.
Yeah, I just want to quickly chip in there. The validations that we've run at, different clinical sites, we have a, central IRB, and there are around, 7 health centers which work with us. And, all the patients that we've seen who have IgM response initially, the subsequent weeks when we collected samples on them, 100% of them actually seroconverted to IgG. So I think that's on the broader spectrum of population, what we are seeing is 100% seroconversion. I just wanted to reiterate that.
Right. I think the only time that probably doesn't happen is if you have an IgG immunodeficiency.
Okay.
So I guess if somebody really thinks they've had it and they don't end up with IgG, then I would check the IgG subclasses.
So total immunoglobulin levels, probably.
Total and the subclasses, the IgG subclasses.
Subclasses.
Mm-hmm.
Great. Let's see. I think we have.
There's also the question of, is there recurrence? And based on my experience with this patient that took, between around 6 weeks to clear for PCR, I think it's just probably, residual viral material. And that, I think probably some people they're just done with the virus in the traditional, 2 weeks, but then some people, the virus does linger longer. So they're not actually getting reinfected. They're just taking longer to clear, based on my experience.
Great. I think we have time for one more quick question. And it looks like there's actually a couple of questions that are similar in scope here, as far as time frame to test. And I know we had a slide there towards the middle of when Hari was talking about the time frame and how we basically collected samples over the course of, I think it was 6 weeks, or maybe a little bit longer. And so the 7 to 10 day or 7 to 14 day window, as far as when the IgM and IgG antibodies were the most accurate, is that from potential exposure or from symptom onset, or what's your recommendation as far as that, Dr. Shippy? Have you seen anything seems to be more advantageous?
Well, it depends on the purpose of the test. Like if you're really just trying to get a diagnosis, I would still do it as soon as possible. If you're just, if you're trying to, see if the person's cleared, you might want to, wait a little longer depending on what their budget is for serial testing. But it sounds like the insurance companies should hopefully reimburse with the super billing.
Great. All right. I know there's a couple other questions out there that we didn't get a chance to get to. We will possibly, what we could do is add a slide or two at the end of these that we can post with the downloadable slides. If perhaps Hari has a few minutes to just give you guys a few answers. It looks like most of them have to do with time frames and patterns of antibodies and things like that. So someone from the lab may be able to give you some answers, since we have to wrap up the webinar for now. But I do want to thank you, Dr. Shippy. Thanks so much for your time today. Your contribution to the data sets, obviously, and our internal studies and things like that, as far as your involvement, and as well as just, your experience with that is, invaluable. And so, on behalf of Vibrant, we're very thankful for your help and your time today.
Oh, you're so welcome. And again, I am so grateful for having this tool to really help guide me with how to help my patients and their families as well. So thank you so much for all this amazing work that you've done.
You're welcome. You're welcome. And Hari, thank you very much for your time today. You spent a lot of time going through a very heavy technological description, and that, I think you're the best person for that. So thanks again for jumping on here and kind of walking everyone through exactly how this process works from start to finish. So
absolutely. Great to connect with the providers and share what we've been doing here at Vibrant. Want to shout out to the team here at Vibrant. They work day and night to bring this test to market. So it's been a great team effort.
Awesome. So thanks everyone for attending. Yeah, the recording will be typically emailed within 2 to 3 days. It'll also be posted in our portal, so you can access it on demand once you log in. The slides are always available for download usually as well in the portal. Any questions, feel free to contact your local sales territory representative. You can also access a lot of this information on our website, vibrant-america.com. And if you click on the COVID-19 tab, it will take you to where you can view a lot of the information that we presented today, as well as if you need to create an account in order to order the test, there is also to do that. You just need to have your NPI number handy. And once you do that, it's a fairly quick automated process. So thanks and have a great day.